My name is Sheri Olson. I'm the Senior Director for the Products, Applications, and Services at Thermo Fisher Scientific. I always wanted to understand cancer in general. I never really understood as a kid why people got cancer. It was one of those things where it was almost like somebody had the unlucky lottery ticket that had cancer. And unfortunately, the friends and family we knew that were afflicted by the disease never ended up surviving the disease. So when I finished my graduate work, I basically wanted to go into the field of trying to ask that question of why cancer, and how can we look at better ways to solve this problem. I specifically got interested in breast cancer research when I landed a dream job at a company studying the hereditary cancer genetics. A lot of groundbreaking research had just blown open in the area of linking genes to families with breast cancer. So my passion, my curiosity got me put on a team of leading scientists, clinicians, and medical doctors, along with genetic counselors, to really go off and study cancer in breast cancer families. And here I thought, I'm actually getting to the root cause of why I became a scientist. The treatment of breast cancer back when I started at this job was really, truly still just experimental. Large part of cancer was dealt with through surgery. And that meant either a single or double mastectomy or a lumpectomy followed by chemo and radiation for women. So working with this team of researchers, we really took a look at BRCA1 as the gene predominantly that had been discovered in 1994 that was linked to hereditary breast cancer. When looking at the BRCA1 gene, our research team really had a eureka moment. First of all, we were looking at actual true patient samples, not just the cloning of the gene. So in looking in healthy cohorts and finding mutations in the gene that could have predisposed or been misread in any way to predict women for cancer, we were able to take a look and saw that there were 7 major changes in the gene sequence out of normal, healthy population of women that had no predisposition to any form of cancer or any cancer in their family back 3 generations. So it was a big eureka moment to see that there are natural changes in the coding region of this gene, and by understanding that, we could now take a look at that and use that as a screening baseline for women and genetic counselors to see if they truly had mutations in the gene. Applied Biosystems technology was really important to this entire process. In fact, coming out of grad school, I was one of the few individuals that actually moved to fluorescent DNA sequencing from radioactive sequencing. And I can say for this particular project, our entire lab used Applied Biosystems, oligonucleotide primers, Taq polymerase to amplify our targets of region. We also used the fluorescent sequencing technology, which was still pretty novel in 1990s. I think it's really important that Applied Biosystems continue to advance its brand to solve key scientific challenges, because it is known as the gold standard of technology innovation. It's been there from the beginning. It's been there through pre-genome. It was there to sequence the human genome, and now it's advancing many different scientific disciplines forward.